the question came is that in a hypothetical trihybrid cross of three loci, that is A, B, and C, all were inherited in a complete dominant manner over their recessive alleles A, B, and C respectively. When a test cross between F1 and parent AA, small bb, and small cc was performed, following genotypes of 8 phenotypically distinct classes were observed with respective numbers. Okay, so this question is basically talking about a trihybrid cross of three loci. Okay, so this is a trihybrid cross, and the trihybrid cross, and they have given the three alleles, the A, B, and C. These are the dominant alleles, and the recessive alleles are the small a, small b, and small c respectively. When a test cross between F1 and parental AABBCC was performed. So what is a test cross and what is a trihybrid cross? A trihybrid cross we will be discussing in the next slide. What they want is that they have given the numbers, means the genotypes, the following genotypes of 8 phenotypically distinct classes. So they have given the number of genotypes among the 8 phenotypically distinct classes. Okay, and their numbers are given in this table, and what they have asked us that the genetic distance up to 1 decimal between A and C loci will be. So they have asked that what will be the distance between the A, from the A allele to the C allele in the chromosome. Okay, so what will be the genetic distance between the A and the C loci, and they have asked us to find that distance in cm. What is cm here? This is not centimolar or this is not centimeter, so this is centimorgan. Okay, so this is a unit. From this unit you will be able to know that this is a question which is coming from the recombinant frequency calculation, and this is a trihybrid cross, not a single cross. This is a trihybrid cross, and what they have asked is to find the distance between A and C. So what should come in your mind by looking into this question is that this kind of means questions comes normally from the recombinant frequency calculations. Okay, so now let us look into what is the recombinant frequency. So this recombinant frequency is actually very helpful in determining, in understanding the linkage of the two alleles in the chromosome. Okay, so this recombinant frequency is given by this formula, that is the number of recombinant progeny divided by the total number of progeny into 100%. So the number that you will get here, this number will come in the unit of centimorgan. Okay, that is your small c capital M. So distance between, so some of the important points while solving this kind of questions of recombinant frequency. So distance between multiple loci can be determined using three-factor test crosses. So why I am means talking about these three-factor test crosses? Because in your question you see there are three factors or three alleles, three genes that we are looking what is their loci and what in which position they are present from one another, means what is the distance between A and B and B and C. If we know that, then we can also calculate that what is the distance between A and C. So this is a three-factor cross, and this is a test cross. Test cross means you are actually crossing the heterozygous parent to a homozygous recessive for all three genes. Means if there are three genes, here there are three genes, that is A, B, and C, and the small a, small b, and small c alleles are the recessive alleles or the recessive genes. So the parent which will comprise these alleles, that is the small a, small b, and small c, this is the recessive parent. Okay, the parent having the recessive alleles. Okay, so if you cross your heterozygous means all these A, B, C, capital A, small b, small c. So this is after the cross. Okay, so if you means cross a heterozygous parent to a parent homozygous recessive for all three genes, then you will come across these three factor test crosses, which is very important for calculating the distance between the genes in the chromosome with using the recombinant frequency, and this is known as the three factor test cross. Okay, so you will know which chromosomes are parental. So from the values that is being given, that here there are these genotypes which are given. Okay, so how to know that which are parental phenotype genotypes or parental phenotypes, and which are recombinant and which are double cross. So here we are coming across a double cross concept. Okay, so there is a double crossover because there are three factors which are present, so there is a possibility that there can be a double crossover also along with the single crossover, just like this. If you see here, here this there is a double crossover, that's why the middle allele that is totally interchanged. Another important thing that you can observe from this numbers is that the parental phenotypes will always have the highest number. So here the parental phenotypes you can identify from this. Okay, so it will have the highest, the most abundant offspring from the test cross. You will know which chromosomes are parental because they will be the most abundant offspring from the test cross. Whereas in contrast, the double crossover offspring will be the least abundant. So see that if you see the numbers, the least abundant are these two. So these two, from these numbers also you can identify these are your double cross. These are your these So these are your double crossovers. That is the DSEO. Okay. Now what is the genetic distance they have asked? Okay. Now there are certain tips that I wanted to tell you, because this kind of question is being asked in several times in the gate. In different years, you will find that they have asked questions from this recombinant frequency. And for a three factor test cross, what you should consider in your mind that there are few steps for solving these questions. First step is to identify the parental offspring. So that will be the most abundant among the all. The most abundant numbers will be the parental offspring. Then you have to identify how many are the double crossover offspring. That will be the least abundant, just like we have seen here. So the parental offspring and the least abundant are the double crossovers. Now determine which locus is in the middle, the one that swaps places in the double crossover. So how you can determine in a quick trick, that what loci means which locus is in the middle. So among A, B, C, it can be possible, right, that the C loci is in the middle of A and B. So B is not in the middle. Okay. So but how you can determine that which locus is in the middle? So for that, what you need to do is that in the double crossover, just check that which one has swapped the places. Which loci has swapped the places. So in the double crossover, if you see here, the B loci in this question has swapped its place in the crossover. So from this, you can know that the B loci is in between the A and C loci. Okay. Now, what you should do is determine the recombinant frequency between one locus and the middle locus. So, then what you need to do is that if it is A, B, and C, then you need to determine the distance or the recombinant frequency between the first loci and the middle loci. Okay, and then again you determine the distance between the another loci, that is for example C, and the middle loci, that is B. First you determine the recombinant frequency of A with B, which is in the middle, and then you determine the recombinant frequency of B with the C, and the B is again in the middle. Then you draw a genetic map, like what is in the front, what is in between, and what is in the next. And then what I prefer that now let us look into the solution of this question, since you now understand the concept of this question, right? So the solution is like this. So what I was talking is that you always need to draw this kind of genetic map. This will help you in solving the questions and also understand what is going on in the solution. What you have to do for finding solution to this kind of questions. So the answer to the question that I have already shown you, and also I have cleared the basic concepts of the recombinant frequency. It is in a 3-point linkage map. The position of the middle marker is determined by comparing the double crossover and the parental phenotype classes. So just like I have said that the middle loci you need to find. So here it is being already I have said that you need to find this middle loci, and you also to identify the parental offspring and what are your double crossover. Then only you will proceed. So the middle marker gets exchanged in a double crossover while the others remain parental combination. So just like what I have seen in the question is that this B is actually changing its place. Then and the other two are the parental format only, parental loci only. Only this B is swapping the place. In the double crossover, how we know it? This is double crossover because this is the least number among the all that is given here. Okay, so that is your double crossover and your B is swapping places. That's why your B is the middle loci. So that is now clear. So by comparison, it is clear that B is the middle marker. Now what I have done is to, for your easy understanding, I have drawn what is happening. So what are the possibilities? So A, B and C, there can be a parental gamete which will completely carry this alleles having this loci, that is A, B and C and small b, small c. This is your parental gamete. Another what can be happening is that there can be single crossovers. Okay, so there can be single crossovers where only one loci is interchanging its place. Okay, but here what you are seeing that this B and C, so if you see there is this small a, small b, small c, capital S, small b, small c. And again in the single crossover, there is another capital S, small b, small c and small a, small b, small c. So these are your single crossovers. But when you have two simultaneous crossovers, that is this cross as well as this cross. So when this can only occur if there is a three factor loci present. Three factors are present. If there are two factors, then the question would be very easy because there will be only single crossovers. There could not be that double crossovers. So here this double crossover gamete will look like this. So now you go back and see what are the means combinations that are being given, the genotypes that are being given. So if you see this genotype and if you now compare what I have written, so they have given only these genotypes, these different combinations they have given. They are different chromosomes, they have given the numbers. Okay, so from here we already know, from drawing also we know, from concept and from looking into the number we know that this is the double crossover. Now what we need to do, the next step is to determine the recombinant frequency, between one locus and the middle locus. So now we know that the middle locus is B. So first we need to determine the recombinant frequency between the A and the A, A and the B, the A gene with the B locus. Okay. So now first we need to determine the A loci distance from the B loci. Okay. So for that what we have done, the distance between the A and C can be calculated by adding the distance between AB and BC. So the total distance between A and C is calculated by just adding the AB distance with the BC distance. Okay. AB, the distance we need to calculate, and the BC distance we need to calculate, just add them and we will get the total distance between the A and the C loci. So now first what we are finding here, this distance between A and B can be calculated by this formula. So what is the formula? I have already given you the formula, that is the number of recombinant progeny divided by total number of progeny into 100%. So here the recombinant progeny will contain the single crossovers for A and B. Okay, so the single crossovers for A and B. So this, where this A and B are involved, the single crossover between the A and B, this one, first single crossovers, this single crossovers plus the double crossover, because in the double crossover also the A and B alleles are involved or loci are involved. That's why the total number of recombinants will be here. The number of SCO1 is the single crossover one, which is this one. Okay, and so you now just need to get the numbers from the table, that is 85 and 80. So 85 plus 80, and also there are two double crossovers, 1 plus 1. So that's why I have written 85 plus 80 plus 2. So here it is clear, right? Means this single crossovers, because you are considering the distance between or the recombinant frequency between A and B, you need to see only those chromosomes or the genotypes where this A and B are involved in the crossovers. So this is this single crossover and this double crossover. Okay, so that will give you the total number of recombinants, and the total progeny is by totaling all the numbers. So you just total what are the numbers which is being given here, you will get this 1000 value, and then now you just need to calculate by means a calculator, you will get the value 16.7 centimorgan. So that is the distance between A and B, but in question what is being asked, the distance between A and C. So now for determining that, what we can do is that we can again find the distance between B and C. Again move to this next point. We need to determine the distance of another locus from the middle locus. The middle locus is B and the another locus is C. So A and B we already found. Now B and C we will be looking into. So then what we will do, we will again look into the table that where this B and C crossovers are involved. So the second one, this SCO2 is this second. Okay, this is here 2, and the double crossover also the B and C is involved. So just total the numbers which is given. So if you go there, if you just look there, then these numbers are given here. Okay, so these numbers are already given here. You just total them and you will get 8 plus 7 plus 2 by 1000, that is 1.7 centimorgan. Now what is being asked, the distance between A and C, and since we now already know that there is A, there is A present, okay, there is this B present, okay, and there is also a C present. Okay, so this is the way. So you now know the AB distance and also the BC distance. So what will be the AC distance? AC distance is just adding AB plus BC. So that will give you 16.7 plus 1.7. The answer will be 18.4 centimorgan. So this is the way how you should solve this question. For your reference, you can follow these two courses of NPTEL. So the first course is Functional Genomics, and the course instructor here is Professor S. Ganesh of IIT Kanpur. And this second course that you can follow is Genetics and Genomics. Here the course instructor is Dr. Josh T. Puthur, and he is from Department of Botany, University of Calicut. Thank you.